Hi everyone, my name is Satria. I'm a PhD in the group of Marix Medema in Wageningen University, Netherlands. And today I want to present about our project about charting the secondary metabolic diversity of more than 200,000 microbial genomes and metagenome-assembled genomes. In our group, we study Bisenacean gene clusters. So those are distinct features of microbial genomes, of which genes that encode for proteins that in turn will act as enzymes and evolve together in a single secondary metabolic pathway are found to be commonly clustered together or next to each other in the genome. And this makes us able to develop computational tools to basically do genome mining to try and predict all the secondary metabolites that an organism may encode in their genome. And given the close link between these genomic features and the actual chemical compounds that they may encode for, we can imagine that if we take all the sequence microbial genomes out there in public databases like NCBI, we can try to do genome mining and then use them to as a proxy to map the biogenetic diversity of all the potential secondary metabolites that those microbial organisms may produce. So here we take around 200,000 genomes from NCBI RefSec along with some 20,000 mugs from multiple published studies, and we predict their BGCs using a tool called AntiSmash version 5, along with some 2,000 BGCs known and experimentally verified BGCs from the MyBig database. And we end up with a total of around 1.2 million BGCs predicted from those genomes. And in order to chart this binary diversity or measure this binary diversity, what we do is we perform large-scale homology analysis. So we group all the similar BGCs into what we call gene cluster families or GCFs. And we do this using our newly developed tool called Big Slice. So you can check out my other presentation in the microbiome courses session for technical details about Big Slice. And we come up with a total of 95,000 GCFs from those BGCs using the default result that we set. And using these GCFs, basically, we can then plot this global atlas of biogenetic diversities from those BGCs. So here what you see is 95,000 GCFs collapsed into 500 bins using the K-means clustering algorithm. And from here, you can see like all the like maps or pattern of the more generic compound classes like polyketides, NRPs, RIPs, or alkaloids and terpenes, etc. But also you can see the distribution of those unique BGCs or biosynthetic potentials across datasets and across taxonomy. For example, here you see on this orange split, BGCs, polyketide NRP BGCs exclusive to the fungal genomes, which are actually pretty distinct compared to the bacterial ones. And you can also use this result to try and measure really how much out there are the underexplored BGCs. So here on these orange dots, you see the GCFs closely related or includes the known BGCs from MyBig. And actually there you see that more than 95% of the BGCs and GCFs out there are still distantly related from any known BGCs from MyBig. And some 3,700 of them, of the GCFs, are actually exclusive to the metagenome-assembled genomes. And one of those BGCs are, you can see here from the Tara Ocean dataset, you can see this NRP polyketide hybrid BGC with a pretty complex multimodular structure, typical of the known BGCs you can see in the MyBig database. And you can also use this result to try and see the basically the most productive species and genus in terms of the unique BGCs and secondary metabolites that they can produce. And here you can see that aside from the frequently sequenced pathogenic bacteria and fungi, you also see the classic uh, secondary metabolic producing species like Streptomyces. And here you also see that aside from the Streptomyces genus, for example, in Bacteria, you see also the Nocardia and the Amycrolatopsis genus having also like a potential to produce a lot of unique TCFs. And in the future, what we imagine is that we can use these gene cluster families to actually replace protein families as a way to do functional metagenomics, basically based on BGCs. And this also in part also be made possible by long-read sequencers able to generate context and reads that spans the whole length of BGCs, which typically like from 20, 50 to some 70 kbs. And yeah, thank you for your attention. Feel free to ask any question.